Principles of Clinical Chemistry Automation. So, history of automated analyzers. So, back in 1957, there was a Technicon. It was a continuous flow analyzer that had a single channel, and it ran samples in a sequential batch processing. Then we moved on to the simultaneous multiple analyzer that was able to do multiple channels, and it was capable of producing 6 or 12 test results simultaneously. And then in the 1970s, the centrifugal analyzers were introduced, and that used centrifugation to help aid in analysis. Automated clinical analyzers. So the DuPont is the automated clinical analyzer option. Was the first non-continuous flow discrete analyzer. It was the first to have random access and introduced plastic test packs instead of glass. Positive patient identification options and required infrequent calibration. Then in 1976, thin film analysis technology came about, and it was introduced by Ektachem, also known as Kodak, but it's now known as Vitros. So first to use micro volumes of sample and reagents on slides. It incorporated computer technology into design and operation. Discrete analyzers in the 1980s incorporated ion selective electrodes, or ISEs, fiber optics technology, and they were more advanced computer hardware and software availability. For modern clinical analyzers, we are always looking for something better, right? So faster and easier to operate than before, of course, improved precision, sensitivity, and specificity. We like the option of a walk-away capability, so automated operation without having someone to constantly do the next step. Instruments available for point-of-care testing. Techniques that have been incorporated include immunochemistry, fluorescence, polarization, nephelometry, and chemiluminescence. These have been designed for higher sample volume and faster turnaround times. Some more trends. We also have fewer test profiles and more individual diagnostic testing. And that was a mandate of Medicare and Medicaid and what they would and would not fund. That kind of helps to influence, what we're doing. There's an emphasis on quality results with high accuracy and precision, and regulatory oversight from CLIA, Joint Commission, and CAP. So, basic approaches. An introduction. So we got automation in clinical laboratories has revolutionized how diagnostic tests are performed. The goal is to enhance efficiency, accuracy, and throughput while minimizing errors in human intervention. So the purpose of getting automation in the laboratory is to increase the number of tests per technician. So automation allows a single lab technician to process more samples and perform a greater variety of tests simultaneously, which improves productivity and reduces labor costs. The second one is minimize variation in results between technicians. So if you think about it, manual procedures can introduce variability due to us being humans. Automation standardizes those test processes, which helps to ensure consistent and reliable results, regardless of who is running the test. Another one is eliminate errors from manual procedures. Once again, human errors in pipetting, timing, and data entry can affect test accuracy. Automated systems can also do that, and that can help reduce those errors. Use of small amounts of sample and reagent. So automation techniques are designed to work with minimal volumes, which is critical for conserving precious patient samples and expensive reagents. So some techniques for laboratory automation is your continuous flow systems. This is where the samples and reagents are continuously pumped through a system. It's efficient for processing large numbers of samples in sequence. And early automation systems like Technicon use this method. And then we move on to the centrifugal analysis. This utilizes centrifugal force to move samples and reagents within the rotating system. It allows rapid mixing and separation during testing. Once again, this was introduced in the 1970s, and this method increased the throughput and precision. And then we move on to the discrete analysis, where samples are processed individually in separate reaction chambers or wells. This enables random access testing where any test can be performed on any sample at any time without having to wait for the batch to be done processing. It supports stat testing, which is an urgent test with a faster turnaround. And this approach is now standard in modern analyzers. So from benefits of the automation in lab, as you guys can probably guess, is higher throughput. So we have the ability to process more tests faster. We have the improved accuracy and precision, which helps reduce variability and errors. Flexibility, which is the ability to run multiple test types on demand. Cost savings, which has efficient use of reagents and staffing. And then enhanced patient care, we get faster results, which will lead to quicker diagnosis and treatment for our patients. So what is continuous flow analysis? Continuous flow analysis is one of the earliest automation techniques that were used or was used in clinical laboratories for chemical analysis. How it works is there is a pumping mechanism. So the reagents, diluents, and patient samples are continuously pumped through a system of interconnected tubing, and there is an air bubble separation in between them. So to prevent mixing of adjacent samples, the air bubbles are introduced between the patient samples. These air bubbles act as separators and can clean the tubing by preventing cross-contamination. So some advantages is it's automated. An automated process is allowed for sequential testing of multiple samples, improved speed and reduced manual pipetting errors compared to the fully manual methods. But there is a disadvantage. The air bubbles help with the carryover, but there is still significant carryover that can occur. Like I mentioned, despite the air bubbles, some reagent or sample from the previous test could contaminate the next, which affects the accuracy. It's also wasteful use of reagents. Since there's a continuous flow, it means the reagents are flowing even when not being used. That leads to a higher consumption cost and use. There is tubing and flow path maintenance can also be pretty complex. So some history behind continuous flow analyzers. They were among the first analyzers or automated analyzers, such as the Technicon system that was developed in the 50s and 60s. Their development paved the way for newer, more efficient automation techniques like the discrete analysis. So centrifugal analysis. This is an automated technique that uses chemical analysis where centrifugal force is utilized to move and contain liquid samples and reagents in separate cuvettes for measurement. So how it works is there is a rotor that spins at a high speed and it generates centrifugal force that pushes samples and reagents outward into individual cuvettes or chambers. So this force separates and contains the liquids within these discrete spaces, which enables the performance of chemical reactions and measurements without cross-contamination. So some key features of this is you can do batch processing. So the analyzer processes multiple samples. Downside is it only runs one test at a time for each sample within that batch. So just a potassium on 10 patients is all you could do. This is distinct from continuous flow or fully random access systems. Some examples that you may run into are the Cobas by Roche or the IL Monarch analyzer as well. Some advantages, it does have precise containment of samples and reagents, which minimizes cross-contamination. Efficient handling of multiple samples with good reproducibility. Now, some limitations is limited to running, like we mentioned, one test per sample at a time, which may affect throughput for high volume labs. Batch processing means you can't usually add a stat sample easily without interrupting the batch, which could, obviously cause trouble if you think about it, right? So some historical stuff on this. So the centrifugal analyzers represented an important intermediate step in the evolution of the lab automation. It helped to bridge the continuous flow methods and to the discrete analyzers. Now to discrete analysis. Discrete analysis is widely used in the laboratory. This is where each sample and reagent are separated in individual containers during the testing process. So how this works, for the separation, every sample and reagent is kept in its own container or reaction well. For the testing flexibility, this analyzer can run either multiple tests on one sample at a time or multiple samples for the same test at a time. So there is random access, and what this means is this method allows random access testing, which means that you can put on a stat sample at any time, and it would not interrupt the ongoing testing that's already going on. So some features of this: it is pretty popular and versatile in a lot of labs today. It supports a wide variety of tests and testing volumes. You may run into some of these when you're doing your clinicals. We have the Dimension, the Vitros, Hitachi. All of these analyzers are discrete systems that offer random access capabilities, and that allows the labs to process the workloads efficiently. Some advantages is it has high flexibility and throughput for various test types and volumes, the ability to add a stat sample without disrupting workflow, and reduce sample and reagent contamination risk. Steps in the automated analysis is pretty similar to the other steps, but we have specimen prep and ID, the measurement of the sample and delivery, reagent systems and delivery, chemical reaction phase, measurement phase, and then we go to the signal processing and data handling. For the specimen prep and handling, sample is properly ID'd and location in the analyzer is monitored throughout the testing. The simplest is manually labeled sample cup in a numbered position, so you can do that by hand, or a lot of labs now will have a barcode that's labeled on it, and it's loaded onto the analyzer, and then the analyzer then tracks everything for you. Specimen measurement and delivery, so there is circular or rectangular racks that hold the samples for you. The cups can also hold standards, controls, and patients, so it's not just patient samples. It can do your controls and stuff as well usually. There is a pipette that is in the reaction chamber that goes into the cuvette. You need to make sure we cover specimens to avoid evaporation. The measurement done by aspiration of sample via the probe, so it goes in and dips into each cup and takes out the amount it needs. And then the probe can also have a level sensing device as well. So if it's a short draw, the probe can detect that before going into the sample. Pipetters may have disposable tips and air replacement displacement syringe to measure and deliver the reagent. Can be liquid or dry for the reagent systems. Liquid is usually purchased in bulk. The dry is usually in tablet form, which is then either lyophilized by the instrument. It's usually like a tablet that's in like a container, and then a lot of time the instruments will go in and add the liquid it needs to make it liquid. In continuous flow, the reagents are from the bulk containers. Vitros uses slide technology, and Paramax uses tablets. The chemical reaction phase in a discrete analyzer, reactants in moving containers. Sometimes those containers are disposable or reusable. Just kind of depends, and you'll know once you're working on your analyzer which one they are, because hand cleaning each of those cuvettes is not a good time. So in the continuous flow, optical readings are taken during the flow of reactant fluids. Continuous flow, there's coiled tubing, so the liquid will rotate and tumble. There are centrifugal analyzers that use start-stop sequences. And then there are water baths as well to keep the temperature where it needs to be. Some of them, body temp at 37 degrees Celsius. The measurement phase, the most common measurement for chemistry analyzers is going to be by visible and UV light via spectrophotometry. They can also use fluorescence measurement, such as fluorescent polarization and chemiluminescence as well. They can also use fiber optics, which help to transport the light signals from remote read stations as well. There are continuous flow uses that flow through cuvettes. The centrifugal analysis measurement occurs while the rotor is rotating, and the slide technology uses reflectance spectrophotometry. For signal processing and data handling, there's the accurate calibration is necessary to make sure that we have the correct results. There are a lot of software programs for calibration data handling that we will use. It also will help to document your quality control recording and linearities for your instrument too. There is also abnormal patient results in your LIS or your lab information system, or even on your instrument as well. Sometimes you can have your patient results be flagged. So if they're, abnormal results, if it found a clot, if the temperature isn't checking out, and examples like reagent volume checks as well. It can tell you like, hey, we're running low. That's when you know to put new stuff on. So total laboratory automation. So what is it? It refers to the integration of all stages of laboratory testing into a seamless automated process, which helps to improve efficiency, accuracy, and turnaround time. So the components of this is your pre-analytical, analytical, and post-analytical. Your pre-analytical, this includes all sample processing tasks before the actual chemical analysis. So an examples would be specimen handling, when you're sorting them out, labeling, when you're centrifuging, and aliquoting samples as well. So really anything prior to it being ran. Analytical, this is the core chemical analysis of samples using automated analyzers. It ensures consistent, precise, and high throughput testing. So analytical is the actual testing part. And then post-analytical is the management and processing of test data after we get the results. This can involve reporting results, quality control, data storage. It can also include interpretation of results as well. So some benefits of having a total lab automation is it minimizes manual labor and reduces human error. It can increase throughput and reduces turnaround time for results. It can enhance accuracy and precision by standardizing processes, and it can also facilitate better workflow and sample tracking. So pre-analytical automation. Pre-analytical refers to, as I mentioned before, the handling, processing of patient samples before they undergo chemical or chemical analysis. So some components of this, use of primary sample tubes. Samples are collected in standard tubes, which are compatible with the automated system. Having barcode readers, specimens are identified and tracked using barcodes, which helps to ensure accuracy and sample identification and minimizing errors. And then you have your front end handling stuff, and that would be your ID and label specimens. So automated systems scan and label samples correctly. Centrifugation, samples are spun to separate components, so plasma from blood cells. Preparation of the aliquots, portion of the sample are prepared for testing or storage. Sorting and delivery, so samples are sorted and sent either to the analyzer for testing or to storage for future use. So some importance of this is it reduces manual handling errors and improves traceability, speeds up sample processing and can reduce turnaround time, and it helps to ensure consistency in sample preparation, which leads to a more reliable test result. Analytical phase. So you can have small sample volumes, and that requires minimal amounts of patient sample, which can preserve specimen, the specimen. Some people you just can't get a lot, and sometimes you need a lot of tests. So it's nice that we need a small volume now. Reduced reagent uses. It uses smaller amounts of reagents, which also reduces the cost and waste. Random specimen placement, so samples can be placed in any order, which also enhances flexibility and workflow efficiency. Expanded on board test menus, so supports a wider variety of tests directly on the analyzer. A quicker reaction time, so there are faster chemical reactions now, and that leads to a faster result for the patient. Same with the higher throughput, more samples can be processed per unit time, and that also improves laboratory efficiency and gives them the result faster as well. Lower dwell time, that reduced waiting time between steps, and it helps to accelerate overall testing. There is user-friendly software for the most part. It is intuitive interface that can help make operations simpler and help reduce errors. We can also have modem connections for troubleshooting. So sometimes if you're having issues, you can have some remote access capabilities with the manufacturer, which can help alleviate some issues that we're having immediately instead of having to wait a day or two for them to be able to come. Integration with online systems, so it facilitates the data exchange in connection with the lab information system or the LIS. So obviously some benefits of this is it has improved efficiency and cost effectiveness. It enhances the flexibility to handle a variety of test requests and better data management and quicker reporting for our patients. And then we have post-analytical. Some features of this is the data management systems. Most post-analytical processes are going to be handled by personal computer-based modules, which are designed specifically for data management. We have software integration. So these systems interface directly with both the analyzer and the LIS to ensure smooth transfer of that data. We have automated quality control management a lot of times now as well. So this software can automatically manage the QC data. It helps to maintain accuracy and compliance without, someone manually having to intervene if needed. Some importance of this is it enhances reliability of the test results through automated QC tracking. It can facilitate faster reporting by seamless data transfer, reduces human error by automating data entry and management, and streamlines laboratory workflow by integrating different systems. Some future trends we got going on. So more integration and miniaturization. Ongoing efforts have been going on to make laboratory components and systems smaller and more integrated for enhanced efficiency and portability. So that falls directly in line with point of care testing. There's been an increased emphasis on rapid testing near the patient. It helps to reduce turnaround times and it helps to improve clinical decision making. Spectral mapping and multiple wavelength monitoring. So advanced techniques are allowing simultaneous analysis of multiple analytes within the same cuvette, which also goes to help improve throughput and accuracy. Mass spectrometry and capillary electrophoresis, adoption of these very sensitive and specific methods help to complement traditional testing, which helps to enable detection of complex molecules and detailed separations. So some significance of this, these advancements kind of help to revolutionize, if you will, diagnostic laboratories by improving speed, accuracy, and versatility. The miniaturization and integration make testing more accessible and convenient, especially in decentralized settings. And advanced analytical methods have helped open up new possibilities for disease diagnosis and monitoring at a molecular level. And we also have robotics and AI. So increased use of robotics for automation of lab processing combined with AI for advanced data analysis, quality control, and predictive diagnostics. And then we have chip technology and biosensors for the in vivo testing. So development of miniaturized chip-based technologies and biosensors that can perform real-time in vivo testing directly within the body or biological systems. So Vitros is one of the options that uses these methodologies. It has a sample cup tray, so the quadrants that can hold 10 samples each. Disposable tips are hand-loaded adjacent to each sample on the tray. There is sample dispensing systems, so the proboscis presses into the disposable tip. It aspirates the sample and then dispenses it onto the slide. The reagent system utilizes multi-layered dry chemistry slides with microscopic layers of dry reagents, which are mounted on a plastic support, roughly the size of a postage stamp. So the reagent slide has 3 or more layers. It has the spreading layer, which receives the specimen, the central layer, which can alter the specimen, and the indicator layer, where the analyte is quantitated by the color developed in this layer. So the slides are warmed up to body temperature, 37 degrees Celsius, and the measurement is actually based on reflectance photometry, which measures the amount of chromogen in the indicated layer. The light is reflected from the bottom of the spreading layer, which passes back through the indicator layer and is detected by a light detector.